Welcome to our third boot camp. Today we're going to be talking with Dr. Noël Burtt and perhaps maybe a guest appearance by Dr. Gaulton, but he is traveling, so it's a touch and go situation. But Dr. Burtt really is, very knowledgeable and deeply involved in the evolution or the start and the evolution of the T1D knowledge portal. It's a really interesting tool that you all should know about, and she's going to walk you through it. I'm going to ask her to do a quick little intro for herself, and when she's done with the presentation, then she will, take some Q&A. So welcome everyone, and thank you so much, Noël, for joining us.
I'm here as well. This is Kyle.
Oh, thanks, Kyle. Thanks so much.
Yeah.
Can you guys each give a quick intro of, sort of just a thumbnail sketch of what, your bio and what you're doing right now in terms of the T1D knowledge portal?
Sure. Kyle, you want to start?
Sure. I'm an associate professor at UC San Diego, and my lab studies mostly the genetics and also genomics of type 1 diabetes. And then I also help run resources along with Noël and the Burtt team to support consortia that are interested in annotating risk variants for diabetes and metabolic disease. Our lab specifically develops a kind of satellite resource to the portal, which is called the CMDGA, which is the Common Metabolic Disease Genome Atlas, which focuses on epigenomic data, other functional genomics data that we then work with the portal team to provide data so that the portal can annotate the function of non-coding variants that affect diabetes risk.
Did you book it for me?
Well.
Yeah, Kyle, that was a great intro, actually. That's a great segue. So I worked with Kyle for many years. My background is human genetics. I've been at before it was the Broad Institute. I came to develop genotyping technologies, the back end of the Human Genome Project, but most of my career has been spent in complex trait genetics, namely in diabetes, type 1 and type 2, building consortia and research groups around the world to try and generate more data to try and understand the human genetics of these diseases. And that has morphed for the past 10 years or so into building resources to make that data accessible. And so that's been my role, particularly at the Broad, doing that for common disease, particularly diabetes. And so I'm lucky to work with Kyle, who's an expert in type 1 diabetes more than I am, but our goal is to make a resource that anchors itself in human genetics. And I'm going to talk to you about that today with Kyle. I'm going to talk about the CMDGA as well too, but Kyle can chime in detail there. So our goal today is really, we participated in the D Challenge last year. We're happy to be back again. As part of the resources that DKNet aggregates and brings together, we hope that this resource will be used as you all embark on the D Challenge. And so that's how I came at this presentation. Anything else, Monica, before I start that you want to do for
No, not really. I think people are, getting up to speed. We have an ongoing communication on a Slack channel like last year, and hoping that we will add you both now after the boot camp, and that you'll be available to sort of like answer on Slack as things may or may not come up regarding the T1D knowledge portal.
Great.
Okay,
right.
Sounds good. Yeah, dive in.
Yeah, throw comments in the chat. Kyle can maybe answer them while I'm talking and vice versa. If not, interrupt me, please feel free. There's not a lot of us in this call, and it's meant to be more of an interactive session really for you all to learn about the resource. So I start with sort of a high-end view in terms of what the resource is oriented and built on, and then I'll give you some tutorial of how to use it and different features of it. So that's the plan for today. So this is the Type 2 Diabetes Knowledge Portal. It's open access to anybody. It's part of a broader resource, which is what I'm going to really focus on today, called the Common Metabolic Disease Knowledge Portal, which brings together a type 2 diabetes knowledge portal, cerebrovascular disease knowledge portal, cardiovascular disease, sleep disorders, and type 1 diabetes into one resource that's built by one single platform that has different views. And we'll talk about that specifically. But the view that you'll hear more about today is the type 1 diabetes view. The idea is it's a single platform, particularly because common metabolic disease have a lot of interactions in the sense that you may want to look at similar tissues, similar genetics, and so on and so forth. So we felt putting it in one resource was extremely powerful. Additionally, you'll hear about the CMDGA, which Kyle referenced. This is a separate sister resource, but they're interconnected, and they're also very useful in terms of different types of lenses by which you'll be looking at genetic and genomic data. So all this is funded and supported by an effort that's a public-private partnership between the NIDDK and 5 pharmaceutical companies called the Accelerated Medicines Partnership for Common Metabolic Disease. This started in type 2 diabetes and has expanded in the last year to include type 1, kidney disease, obesity, cardiovascular disease. And the idea of making this data, genetic and genomic data, accessible, but also ideally to develop new targets for these diseases. So we come at this from human genetics. And it's a challenge, right? Because over the last 15 years, really what's happened in the field of human genetics is we've identified a lot of regions of the genome, hundreds of thousands at this point, associated with common disease. So it creates kind of like well, I guess everything's associated in the human genome. But ideally, our goal is to figure out how to tease all of these down to function, to ideally find what is the variant, what is the mechanism, and what can help potentially treat patients with this disease, ideally. So I'll talk a little bit about type 1. So these are slides courtesy of Kyle. He can explain them better than I am, but I'll quickly go through them, which is, for type 1 diabetes, to date, about 136 independent signals have been associated with type 1 diabetes. About 90 loci are contributing to over 30% have independent signals. So the challenge here is there are a lot of signals. We don't know what they all do, right? And even more so, if you were to, fine map and determine which is the likely causal variant. So if you do a genome-wide association study and you whittle it down to a region, in many cases, you're still going to have a large set of variants that are contributing to the signal you're seeing from the statistical association data that are contributing to the signal at this locus. So the challenge is 90% of these map to non-coding regions of the genome, even if you find a single variant. So the challenge is, what do they do, right? And each signal has an underlying mechanism by which the risk variant contributes to the disease. And that can help you need to reveal the cell type, the gene, the pathway underlining that mechanism. So our goal is to understand this mechanism of these signals, and you can do this by getting detailed annotation of the genome along with the human genetics. So this assumes a model. Everything that this resource does assumes a model starting with genome-wide association, statistics association. And the assumption is genes contribute to cellular function, contribute to organismal function, right? The idea being that those are regulated by regulatory elements into cell types and states. The idea is if you change something in the cell type of the state with a variant, that could thereby impact the cellular function and the organismal function. So that's this model that underlies the entire resource that we've built here. So the idea being, for many of these studies that are now mature and have identified all those regions I told you in the earlier slides, it assumes you get a region. Well, the challenge is, how do you go from here to a mechanistic approach? This is a nice example from Lena Klesnitz et al., who determined a mechanism at the FTO locus, which is associated with obesity. So here, there are many key questions you must ask, right? What is the variant? What is the regulatory effect? In what tissue? What gene? Mechanism, pathway? It's complicated, right? Well, our goal is to ideally create a resource that helps you do that. Because right now, if you were to do this without this resource, you'd have to go to, say, a website. So here's an example. Like if you wanted to determine all the independent signals for type 2 diabetes, you'd go to a website and you download the GWAS data. You'd have to know how to deal with GWAS data. You'd have to know how to get the credible sets, which I mentioned, which are the signals that we believe are the most causal signals contributing to that signal you see at that region of the genome. You have to pull that from some auxiliary file somewhere. You'd have to go collect data specifically in those tissues and that disease to try and determine what is the actual mechanistic effect going on in those tissues or cell types. You have to overlay those annotations with the genetics to see what's going on there. And then ideally, you have to link the genes to the variants. And this is a large process. Many different resources do this. Many people all over the world are generating data on this. And then if you get lucky, you can embark on a functional study, right? Well, our goal with the type 1 diabetes knowledge portal is to facilitate the process by cataloging all these data and all these resources and make them accessible in a way that is not for the expert, but someone who actually is studying a specific part of the field and may be able to, interact with other disease data sets that they may not be experts in a way that's intuitive and interactive. So what are the foundational data sets? I talked about this briefly, but just to summarize, genetic associations, functional genome annotations, and then bioinformatic methods that we run on all these data to produce new results for you. So for genetic associations, really to remember here is these are GWAS summary statistics for relevant diseases and traits associated with common metabolic disease. We have extensive phenotype data. And we also have things in all ancestries. The ideal is to get, across the entire globe. Functional genomic annotations. These provide, predictions of the variant, the effect on the cell type, and the regulatory elements. So these are independent data sets that marry with the genetics. And this is generated all through Kyle's resource that he was alluding to called the Common Metabolic Disease Genome Atlas. And for this, they have a host of data sets that largely right now represent 4 major categories: cis-regulatory elements, gene target predictions, variant allele effects, and gene activity and function type annotations. And there's over 4,000 in them, and they're enriched in tissues that are common metabolic disease oriented. And I'll show you some of this in a minute when I get to the resource. And then finally, if you have genetic data, genomic annotations, you need ways to integrate them. Many of the methods that are out there integrate them to make hypotheses about the functional consequence or how molecular or cellular function going on at these tissues or these regions. So our resource has about 12 methods implemented at scale across all data sets and diseases, some of which are published, some of which are novel, that allow you to see results from these predictions. They include calculated credible sets, meta-analysis of all the summary statistics in the world for genome-wide association studies, prediction of variant effects, tissue-specific annotations, co-localization, and so on and so forth. The idea is the resource is constantly updated with the best practices in these methods as we're bringing in more data to continually refresh and sustain the resource so it's consistent. And so this all comes together in one single platform that brings these data sets together and ideally makes them open access and integrated through visualizations. So let's talk about the resources a little bit. So when you go to the homepage, this is what you'll see. And you can select disease-specific portals if you're only interested in one lens. So I mentioned the 5 different portals. The type 1 diabetes is the one we'll speak about a little bit today. So if you click on type 1 diabetes, it brings you to this page. It's exactly the same resource. It just whittles the traits and the diseases down to things specific to type 1 diabetes. But at any point, if you're interested in going back out to the common metabolic disease resource, you just click here and it expands to all the diseases and tissues and annotations relevant to common metabolic disease. So first thing you might want to do is understand what data we have. So if you go there, you go up to the genetic association tab, and you can see all of the data sets we have, all the data types. This is for the genetic data only, organized by phenotype group. And if you wanted to peruse through this, essentially what would happen is if you clicked on any of these, you'd see the detailed phenotypes that we have for each of these that are curated. Here's an example for diabetic complications. What's lovely about this is these are phenotypes contributed by folks in the community who have curated the trait and analyze it in the way that they think is best for that disease area. So you get these really well curated, well phenotyped data sets for particular phenotypes that may be really interest to people who are studying complications. So this comes from folks that understand complications. And below the fold, what will happen is the resource updates to tell you what data sets we have for all these particular phenotypes. And if you want to go into these in detail, we have all the provenance, all the citations, all the descriptions of the data sets relevant to that particular a collection in the portal. So you can have that sort of documentation. So that's sort of what is in there. That's sort of the access to the genetic data. So if you wanted to start with, what is known genome-wide about type 1 diabetes? So you go to the phenotype tab, you type in type 1 diabetes, or you could type in any phenotype that you're interested in that's associated with type 1. And you get what we call, the Manhattan plot and the QQ plot. This essentially is the summary of all the genetic association data we have in the knowledge portal for type 1 diabetes samples. And the top of the page starts with the summary. Right below it gets you top associations. So these are the top single variant associations for type 1 diabetes with their p-value, direction of effect, the closest gene, and the consequence. And what's nice about this, if you are computationally savvy, you can download this right away into a CSV file to do analysis on it. I'll show you other ways to get access to that, which may be of value to you as we go forward. But this is the top table. You can sort and filter it by your favorite gene or p-value to see the table automatically update. What's cool about this is that if you've ever used like the genome GWAS catalog or any other GWAS atlases or things like that present GWAS data, they often just give you study-specific results. What's unique about the Knowledge Portal is that we do a meta-analysis that accounts for sample overlap. So it's a very cool method that says, like, because many studies include the same samples because they're getting bigger and bigger and bigger, right? As they're collecting and collaborating to get the largest GWAS for type 1. So for example, the last GWAS for type 1 that we got included, 90% of the samples that were in the last one that we integrated. But you don't want to double count those. You want your user to get the best bottom line p-value that we know about for this particular ancestry and this particular trait. So what we do is we do an overlap-aware meta-analysis, present those results to you at the top of the page that I showed you. But then if you want to understand the data sets that went into that, that's listed here for you. So you can see exactly how many samples, what ancestries we have, what data sets contribute to that. Why this is masterful is that, other resources don't do it. So you'd have to download all the GWAS statistics and then do a meta-analysis yourself. But also what's lovely about this is this does things like gets rid of spurious associations and also uncovers new ones. So this is some work that we've been doing for all of our traits, and it's an unpublished method, but we've done some really excellent validation to see that it really seems to be very, very, very useful. The other thing you can have are gene level associations run by a method called MAGMA. This tells you what is the top common variant gene level association for type 2 diabetes. Again, this is filterable and sortable, and it's on the same page as the phenotype page. And then tissues that are globally enriched for your particular trait. So this is of interest if you want to start thinking about genomic annotations or tissues that may be, associated with your favorite trait. So that's the phenotype page, so at a glance. The next pages are particularly, I would say, more of interest to you all, which is, I want to query a gene, a region, or a variant. So I have my favorite gene, region, or variant. So you type that into the search bar, and you get the top of the page defaults to the associated region. So it may include multiple genes, right? Because in a GWAS association, you don't just get, one SNP, you get a region of SNPs associated. So it defaults to that. And so that's what you'll see at the top of the page. Then you'll see what essentially a PWAS plot, which essentially says for all of the traits we have in the common metabolic disease knowledge portal, what are the genome-wide significant associations? So for this, it's actually, enriched with blood traits. But what you see over here is type 1 diabetes for the BAC2 locus. And then that's the top of the page. On the bottom page, and then there's one other view if you want to see a summary of this by phenotype group. So these things you can click through and sort of see the details. Right below it, it gives you the lead variant for that particular trait. Typed in this case, we're seeing actually blood cell counts is when I defaulted to. But if you can switch to whatever trait was listed at that table right above that I showed. And it shows you the top variant and also all the variants that contribute to the signal if you wanted to know that. And the direction of effect, again, all these are, again, downloadable and accessible. At the bottom of the page, you get the variants and region of the top traits. This gives you a detailed view of the variants that contribute to the association of that closest gene, p-value. And you can also combine multiple phenotypes and see the direction of effect. So for this, I'm showing type 1 and a blood cell count as well. This may not be of interest, but this gives you an idea of the type of searches and queries you can do. If you want to search your favorite variant, you type in your favorite variant, you'll get the associations and all the variant consequences and a PWAS plot of the association across all the traits we have. Again, this is filterable, downloadable, and accessible. You can also see this in a forest plot, which is the more classic view you see in many papers. So if you want to prioritize, if you're really interested in a locus and you want to layer on different types of information and you want to do it from a visual perspective, at the bottom of our gene page, and our region page is LocusZoom, which some of you may be familiar with if you're a geneticist. It's a way of visualizing a region and association statistics. What's nice about this is you can start to add in custom features and annotations and tissues so you can see where your variant may line up with relevant tissue annotations. So I'm just going to briefly walk through this one because I think I have a couple more things that will be more interesting to you. But it allows you to search your favorite variant and set it to that locus. And you can also see the most significant association there by highlighting here. Then you can pick your favorite tissue, your favorite annotation, and a credible set, and the table updates below and allows you to overlay different things. What I love about this feature is the ability to actually link it to the underlying data. So for here, this particular variant overlaps with an annotation that is very
Oh, Noël, I think you froze for a moment.
data, relevant publications and whatnot. And I think that's really lovely. And this allows you more detail about the supporting annotation. And this is a little bit look into, hopefully you're hearing me. My internet connection just went down. Are you guys still hearing me?
Yes, yeah, it went out for a minute, but you're back.
Sorry. All right. So Then one thing I want to just take a quick detour into a new resource we're building, which is right here. If you see at the top of the page, you go to the variant page, you'll see prioritize variants and region. This is a more, more of exploratory visualization of a given region. And I'll just quickly sort of walk through it for an example for type 2 diabetes. It looks very similar. What it allows you to do is add in all of these same things that I showed you above for the credible sets and annotations, but it filters the table down dynamically and then allows you to export them. So this would be like, let's say you have a very large region, you have no idea which variant you may want to study or move into functional assays, but you want to make some hypotheses based on credible sets and annotations. So this is a resource that actually allows you to do that with all the data.
Can you guys, Noël, can you show this with T1D for them right now, or is it still in building, in progress?
I can show it, I can try and show it afterwards, but it wasn't as illustrative for T1D because our newest release will probably have some better stuff for T1D.
Sure, okay, sure, no problem.
But this is definitely, this gives you the functionality. So if you see the credible sets, you can select your favorite credible set and it will upload. And what it does is, what's cool is the table below filters down to just the variants thing included in the filtration you're making up ahead. And then if you want to add your favorite annotations, it gives you a visual of those annotations that overlap with that credible set. And then what's really cool is it allows you to then see, based on bioinformatic methods, we have gene linkages based on those annotations and those associations. And what's, oops, I can't show you the bottom, is right here at the bottom, it filtered down to just one variant. And this variant, if you clicked here, will take you to the evidence in CMDGA, allows you to see exactly what contributed to that. So this is a very new resource that will come out with a brand new version in October, end of this month, which will have way more data and be far more useful to you. So it's coming. And if you want any questions about it, it's on the portal now, but it's going to be well updated really soon. And then, so one sort of other way of viewing our results and data is if you are not, you don't really like using UIs and you're really computationally savvy, for any page in the portal, if you go to the top page, you'll see this little cloud download. This is called Lunaris. This allows you as a user, for whatever page you're on, to download all the table, CSV of all the different results from each of the modules for that given region that you've queried here. Or if you go to the line below, it says open Lunaris to get more information. You go to Lunaris and you can custom type in whatever regions you want, select from our annotations, select from our bioinformatic methods to actually get a download of all those results directly to you. We also use this is all for the API-based way of having programmatic access to the resource. These are sort of two very different ways, which I think might be valuable for many of you working on, integrating results from various resources for type 1 diabetes. So I'm going to actually, before I stop, I want to take you to the resource and show you some of these things in real time. So a couple places to find things. And one more thing I want to tease with you is if you want to find the variant sifter that I was just showing you, that's right here. But I'll take you there and allow you to search your favorite gene and region and do all the things I was mentioning before. Another thing that's probably the most exciting, I would think, frankly, is our predicted effector genes. So this is a sort of a summary of all the information that we do here, which is so many of these GWAS papers now too are also trying to do this. They see their top loci for their given trait, but it's not enough to really do that. What you want to do is present to a biologist or someone who's in industry who wants to have a short list of targets they might put forth for functional study. You want to give them a summary about the gene. You really don't want to say like, this locus. You want to say like, we believe this gene is the effector transcript at this particular locus. So many researchers are doing that now. And what they're doing is linking all the relevant data that I just talked about here, but creating these effector gene list curated summaries. Well, what we have on the resource now is a way to get access to those curated by the community, but also integrated with all the human genetics and genomic annotations that we have at our disposal. So coming in October, we're going to have these for about 40 diseases. Right now we have about 12. We're doing this comprehensively across all common disease, but one of the ones that I think is extremely useful, particularly for you all, will be the type 1 diabetes effector transcript list that we have from Kyle's team right here. So if you go here, what you'll get is a gene-based summary of the genes we believe are the most likely effector transcripts for type 1 diabetes and all the relevant evidence sources that went into that prediction or that assertion. And these are tiered by a different ranking system that's all described up here in the research method. But what I love about this is sort of like this directly allows you to sift down through all the stuff that you might see in the resource to just the facts, ma'am, so to speak, right? So if you here, you'll see the evidence sources, you'll get all the different types of evidence that they used in their weighting. And moreover, if you want to actually get to that evidence directly, you could click on this and it brings you right to that evidence source. So there's this whole like detailed provenance and allows you to drill down to those results. And this is also exportable. So this is something you can pull right out and use in any other downstream computational or integration that you want it to do. So let me just pause and show you a little bit more about that because I think that's really valuable. So if you wanted to, like, let's say CTLA4, you want to learn about that gene. Again, it's integrated with the resource. So this will take you to the Common Metabolic Disease Knowledge Portal entire window for the region page. And you can see all the information we have for all of common metabolic disease, not just type 1. So it's a way of seeing both sides of the coin within the resource. So we're going to have another one of these coming out in about 2 weeks' time as well. Another effective prediction algorithm from another group that we're going to pull the two of them together and compare and contrast them on the site with all the same information. So that'll be another resource available to you to make some decisions and hypotheses for the D challenge.
Oh, that sounds great. Where's that resource coming from? What lab?
That's going to come from Steve Rich's lab, actually.
Oh, yeah, I just saw that he had that paper. That's great.
Mm-hmm. So we
Very
worked with them to get that integrated because they help build the portal as well with us. So
Mm-hmm.
our trusted colleagues as well.
Yeah.
So again, any information about the policies, how we use the resource, if you want to contact us right here, it comes to a very small group of us. We get back to you within 24 hours. But again, we also have the Slack channel for the D challenge, but that helpline is really good too. Again, there's other resources and information if you're interested in other things, but that's sort of the broad summary of the resource. And I'd be happy to take any questions or walk through anything in more detail.
That was great. Super presentation. Thank you so much. Is there anyone in the audience who's interested in asking a few questions? I guess I'll ask a question. So how do the resources here compare, contrast to those coming out of Penn in the DB-GAP, in PANC-DB, and maybe, those available sort of at the I receptor group coming out of the Antibody Society? Do you guys have any thoughts about that? It looks like you have a lot of information on the non-coding regions of the genome, right? That's really where it sits. Whereas I think maybe some of the others may have sort of coding regions.
We have both.
Yeah.
But in terms of the making sense of GWAS loci, a lot of times, your variant's going to sit in a non-coding region. Therefore, that's why the, getting genome-wide annotations helps whittle that down and make decisions about, what gene might be affected by that association and what tissue and whatnot. But to your point about the other resources, so I think like NPOD and HERN, some of the data there, because we're all NIDDK funded in many cases, our goal, and Kyle can correct me if I'm wrong, is to integrate the relevant data that's in those resources in CMDGA and sort of bring those all into one hub that can be married with the human genetics data in visualizations. I think that's one of our goals specifically. Kyle, do you want to comment on that? Did I get that wrong or anything like that?
No, that's right. So PANC-DB has data generated by HugeAMP, which is specifically focused on the pancreas and annotating like clinical features of the samples that they had, but then they also have genomic data like gene expression and stuff like that for a lot of their samples. And it's also non-diabetic, type 1 diabetic, as well as type 2 diabetic individuals that they have data from. And on the genomic side from PANC-DB, we do actually share data. So they send data that they generate that's relevant to our resources to us. So we have data from HugeAMP that's in the genomic space, again, like gene expression type data in our database that then we use to put forth to the portal to annotate variants. So there is some integration of these resources to make use of data in various channels. Those resources are less focused on human genetics, whereas the portal's like primary focus is on annotating risk variance for disease. So there are other aspects of those resources that are kind of independent from our goals of annotating variants, but we do try to work actually quite closely to share data between the resources.
How about like someone like Tim Tree with the UK Biobank? You know, I know he's gathering a lot of data there. Is there a collaborative piece with him and that group? Are they adding value to the T1D knowledge portal?
I don't know. Maybe Noël knows. I don't know what they're doing. And I mean, obviously, UK Biobank is a massive, but it's something
Massive, but
specific.
he's specifically T1D, yeah, and he's been. My understanding is he's been creating a specific database from that resource. So I didn't know if you had been in contact with him, but yeah, I think it really this resource that you guys have built and are currently, you know, growing is a really excellent resource for all participating in the D challenge. And I would encourage you guys to really start playing around with it this weekend and look at it, dive in, and get familiar with it. And I'm gonna, Magdalena has a question.
Yes, thank you. That was a really great resource. I had a chance to look at the website a few days ago, and I'm particularly interested in looking at epigenetic changes. And I think I was able to find some ataxic data and DNA methylation. But if there is some good and easy way to look at specifically type 1 diabetes epigenetic data, if you could point me to that, that would be great. But I also have another question regarding epigenetic, and that is, are there studies available, or are there plans for studies looking at epigenetic association data instead of genetic to look at epigenetic changes, if we believe that many of the changes occur at enhancers rather than SNPs looking at epigenetic data?
Good question.
There's there's depends on the tissue and depends on the but yes, I mean, we have a fair amount of data now in our site, the CMDGA, that in some contexts gets passed forward to the portal, such as chromatin QTLs, so variants that affect chromatin accessibility, histone QTLs, variants that affect histone modification levels, some DNA methylation QTLs, I believe. So we have some studies that have mapped genetic effects on epigenetic changes, and those types of data sets are increasing, giving currently funded studies that would be giving data to us within the next year or so. But we do have some of those data on our site now. If you want to go access the results from those studies directly, our resource, cmdga.org, is probably the easiest way to go grab like the data from those studies. But like I said, in some cases that information is also incorporated into the portal. If you want like the summary stats or something, or like the results of those analyses or whatever, then those data sets are in the CMDGA, which is our epigenomic site that passes data to the portal.
Can I further that question to who is doing the most work right now, or what group is doing the most work right now in, you know, kind of mapping the epigenetic changes in the prodrome of PBMCs, in, you know, the prodrome of type 1 diabetes? Is anyone? And is that data readily available?
Mapping changes in blood from non-diabetics and type 1 diabetics?
Yeah, epigenetic changes specifically.
Yeah. I mean, Steve Rich's group had a paper where they did GWAS. Sorry, not GWAS. It was targeted association study, so it wasn't genome wide. And they also had it wasn't bulk PBMCs. Was, I believe, sorted CD4 T cells, where they had chromatin profiles from a large number of samples of sorted cells, and I believe that was non-disease as well as type 1 diabetic, and so they have they map variant effects on CD4 T cell chromatin in those groups.
That's specifically though for at onset. Has anyone looked at like 1 antibody, 2 antibody in the progression to diabetes, the prodrome?
Well, we're doing that in the
Good.
pancreas. So we have actually about 100 samples that we have data for from pancreas samples from NPOD, which is a type 1 diabetes specific biorepository for those who are not familiar. And all 100 samples are some range of no autoantibodies, 1, 2 autoantibodies. And so, yeah, we are actually just starting to look at those data now. So that would hopefully give us some insight into changes that are occurring, particularly for the 2 autoantibody people that are at some point would have developed type 1 diabetes, almost certainly. Changes that may exist at a state prior to type 1 diabetes. HPAP also, and PancDB also has a fair amount of genomic data from non-autoantibody individuals as well as people with multiple autoantibodies. They don't have genetic data that they provide, I think, but you can certainly use the resources from PancDB to look at genomic changes in the pancreas and non-autoantibody compared to single and multiple autoantibody individuals. That's in the pancreas. In the blood, I know that, well, the NIDDK funds resources through like TrialNet to do these types of studies. I don't know if the data is available yet, but I know there's some funded studies from TrialNet cohorts where they're looking at these same questions in PBMCs. And I think those data, if they're not available yet, would at some point be available in the central repository, which is like the NIDDK repository for large data sets. And I don't believe that that's available at the moment, but once it is, that would be a priority for us to use those data as well. There may be other groups that are doing those studies in PBMCs. Trying to think.
How quickly do you guys does the knowledge portal access and pull down these data from the NIDDK repo?
Not so often. I mean, most of the data we get is either from publications, where like somebody will publish a study that we believe has relevant data, and so we'll either grab the data that in like Geo or dbGaP or something, or, and then deposit ourselves, or in some cases we'll work with the investigators who wrote the paper to deposit the data, as well as since Noël mentioned, this is funded through a large consortia data that is coming from consortia members that are funded through various channels. Data like I was just talking about will exist at some point in the central repository. I don't believe so. But like I said, I don't think those data of like looking at changes in PBMCs of autoantibody positive individuals, I don't believe that is there yet. I think that's an ongoing effort still. But yeah, I mean, we generally try to stay abreast of everything that's relevant from many different channels, NIDDK repository included, that we think would be of value, and the amount of time it takes once we identify data sets of interest, depending on how long it takes to get approval to access the data. Post that, it's probably like, I don't know, like maybe at most a month for us to then deposit data in the site, and then there's a bit of lag time for the data to then be passed on to the portal. So probably a couple months in total from deposition to being available in the portal.
So would you suggest that maybe people look around in the NIDDK repository?
Yeah, I mean, there's a lot of resources in the central repository that people might find useful. And I'm trying to think. I think there's some older studies of looking at PBMCs, but I think most of those older studies are looking at non-diabetic and type 1 diabetic. So disease has already happened. So I think the current focus on looking at the subset of people who are, likely would have developed type 1 diabetes at some point in the multiple antibody space, like that focus, I think, is more recent. And I think a lot of those large genomic studies that are profiling that set of people is ongoing. Like I said, us in the pancreas, as well as Klaus and HPAP, and in PBMCs, I think there are a lot of those efforts are either through TrialNet or other investigators like Steve Rich.
Great. That was a very thorough answer. Thanks so much. Thank you again for this really great talk, very informational and very helpful. And we will see you guys at the next boot camp. And thanks again, Noël and Kyle, for taking time out of your day. Appreciate it very much. Talk to you later. Bye.
Thanks, guys. Bye.
Bye.